It's now time for our last speaker, Alan Rodrigo from Nescent. And I should say that, as you probably know, Alan and Nescent have played a very important role in furthering the development of the field of evolutionary medicine and the creation of the society. So it's really a pleasure to have him here speaking. Thank you.
Test. Is my mic on? Oh, good. Thank you very much. That's a great introduction, and thank you to the organizers for giving me the chance to speak on this work, which is actually a new direction in our lab. I'm a computational evolutionary biologist, and until maybe about 5 years ago, spent most of my time working on virus evolution and phylogenetic methods. And then we became interested in microbiomes because everybody else was becoming interested in microbiomes. And so most of this work, I should say, is done by my graduate student, Shenlong Zheng, who is unable to be here, and my two postdocs, Stephen Wu, who is actually here at ASU now, and Jeet Sukumaran. So I just want to preface my remarks by saying two things. When we think about evolution, it's natural to think about natural selection and adaptation to explain variation within populations and the diversity of life writ large. However, neutral models, the idea that you can achieve change and variation and diversity through stochastic effects alone, have been a key part in the development of evolutionary theory, at least for the last 60 years, but actually dating back all the way to Fisher and Wright when they were thinking about things like genetic drift, etc. So the other thing that I want to say here is to talk about models. And when I think of, as a computational evolutionary biologist, one of the things that we do things like analyze data, we do things like develop algorithms, but we also do things like modeling. And a natural question is, how does one begin to model? And there are a couple of approaches to doing this. There's the kitchen sink approach, where you put everything in, and there's the other kind of approach, the minimalist approach, where you start to develop models from the ground up with a minimal set of assumptions. And that's what I'll be talking about today. So you've heard a little bit about microbiomes. I don't need to go into details here. They're implicated in a range of functions, and they've been found in animals, plants, most organisms that have been looked at. In humans, of course, they've been associated with a range of health outcomes: allergies, obesity, anxiety, and depression, asthma, and other respiratory illnesses. So there's been an explosion of research. It's fair to say that most of this research has been experimental, and I don't mean that as a derogative thing. I mean it to say that we've been testing hypotheses using experiments, but there's been not a lot of formal theory. And when I say formal theory, I mean quantitative theory that has driven our understanding of the ecology and the evolution of microbiomes. So our goal really is to start to think about how we might develop this framework for understanding the evolution of microbiomes, and we begin with a neutral model. And our questions are simple, right? So we want to understand at the start what patterns emerge as a consequence of stochastic effects only. And what can these patterns tell us about the likely drivers of microbiome evolution? If you look at all of the things that you were to list, if I were to ask you to list the factors that influence microbiome evolution, you would come up with a list that looks something like this, and perhaps add a little bit more. You would talk about random chance, you would talk about competition, you'd talk about host genetic effects, the ability of microbes to live in different environments. You talk about microbe interactions, and you'd also talk about heterogeneity, both in terms of the host environment itself, the different body sites, etc. So you could build a model with all of these things in, but then when you do so, people might ask, well, what happens if you leave this out? Or what happens if you leave that out? So really, our approach is to start with the minimal set of assumptions, and these might be the ones up the top here, right? Neutral processes. And when you think about these neutral processes here, you might also have to add on certain requirements that are just necessities of the model themselves. So the neutral processes that we're talking about here, random chance and sampling effects, initial colonization success, competition for space, host phylogeny, those are natural natural things that you might want to have. And then you need to think about how individuals acquire their microbiomes and and where these microbiomes come from, the environment. So we start out by asking exactly those two questions. We have to model two things. As you evolve, as you move from one generation of hosts to the next, where do those individuals acquire their microbiomes? So that's the first thing. Acquisition has to be the first step in modeling microbiomes. And of course, the natural answer here is that they could come from the parents, or they could come from the environment, or a mixture of both. But as soon as you say they could come from the environment, you then have to model the environment as well. And you have to say, well, where does that environment come from? And you can imagine, again, a mixture of two processes. You could imagine a fixed environment that is constantly being replenished with microbes from external sources. Or you can imagine an environment in which the hosts themselves contribute microbes to that environment, and a mixture of both of those. So if you think about it, those constitute the necessary minimal processes that you need in order to model this microbiome. So here's an example. Here's a pure parental inheritance process where the parents give everything to the offspring. Here's the pure environmental inheritance process. Only the environment contributes to the offspring. And here's a mixture. Right? And with a mixture, we can specify in our model how much comes from the environment and how much comes from the parent. Similarly, from the environment itself, we can think about the fixed environment, which is replenished at each generation. We can think about a mixed environment, which has both the parents contributing to the environment and the environment itself replenishing. And we can think of a pooled environment where only the parents contribute directly to the environment. And this last slide, that's an interesting pixel. It doesn't. It's supposed to show a genealogy, and on my screen it does, but it doesn't show it here. If you squint, you can see what looks like a tree. You can see a tree over there. The point that I want to make here is that what we are doing that is different from what ecologists do when they think about neutral theories of biodiversity, etc., is that the spaces that our microbiomes inhabit, the hosts that our microbiomes inhabit, have a genealogy. They have a history. And that history leads to the extinction of certain lineages and the persistence of others. In contrast, in ecological modeling of biodiversity, we have an indefinitely existing set of spaces that can exist. So, bearing in mind that lineages go extinct in genealogies, we can imagine that these will shape the eventual outcome of diversity. So our simulations, we simulated under a variety of values of different ratios that come from the environment, that come from the parents, etc. You look at these numbers and you'll say they're laughably small. That's true, and that's because of the time it takes to do these simulations. But we have also done simulations with much larger numbers, and the patterns remain the same. And what are we measuring? We're measuring relative OTU abundances, the taxonomic abundances. The common measures of diversity, the alpha diversity, which is measures of diversity within a host, beta diversity, which is the diversity between pairs of hosts, how many species they share in common, and gamma diversity, the diversity in the entire population. Now, I should say that we started out our simulation, so we simulate forward in time. We have host populations at the start, and each generation is a discrete generation. There's no selection acting on the hosts, so the hosts are being sampled randomly. It's an aspect of neutral models that everything is mediated through sampling alone, through stochastic effects of sampling. And so, as we move forward in time, and our hosts are sampling from these populations, we have started with a uniform distribution of microbes. Anyone who's measured microbes knows that there are no uniform distributions of microbes. So why did we bother doing that? We did that to ask a simple question. Could we get a skewed distribution of microbes emerging as a consequence of these neutral processes? And the answer, of course, is yes. Yes, you can. So if you have sufficiently high values for parental inheritance and parental contribution to the environment, you can get skewed distributions. And here we've been able to fit these skewed distributions to both the log normal distribution, which is very commonly used, and another kind of distribution called the Dirichlet multinomial, which is actually a very good distribution for modeling. So the answer is yes, we can get these distributions. And what patterns of diversity emerge as a consequence of this? So these are the patterns of diversity that you might get. So here is alpha diversity. Here is gamma diversity. So alpha is within the host, within the population, and there is beta diversity. I draw your attention to this axis here because you'll see that it's actually on an exponential scale. And the reason for that is because at low values of parental inheritance, where a lot of things come from the environment, alpha diversity remains high. The environment is constantly feeding in microbes into the hosts. Makes sense. But as you increase your parental inheritance, your alpha diversity decreases. So it becomes fewer and fewer species remain in the population over the evolutionary timescale that we're looking at. The same is true for gamma diversity. Beta diversity has an interesting nonlinear relationship with parental inheritance, and the reason for that is simple. When you have very low parental inheritance, most of the species, the same species are being collected from the environment. So every host tends to have the same species, meaning that they look similar. When you get very high parental inheritance, then only a few species remain, and they all come from the same common ancestor of hosts. And so again, they look very, very similar. And so only in that, there's a sweet spot there where you have beta diversity being relatively high. So if we think about this, what are our take-home messages from this neutral model? The less contribution by host from generation to generation, the high average and overall microbial diversity. And that makes sense, because the environment is adding and replenishing those microbes in the hosts. The interhost diversity, as I said, has this nonlinear relationship with parental inheritance. So if we want to think about this in relation to real data, you have to think about it from a comparative perspective, right? And it's hard to come up with ideas on how one might want to compare this, because of different species have different relationships to their environment, they have phylogenetic constraints, etc. So it would be nice to have pairs of species that actually have, where you know there's more parental contribution or less parental contribution. And lo and behold, the nicest species to think about are bees and wasps. So bees and wasps, you have solitary wasps and bees, and you have colonial wasps and bees, social wasps and bees. And it turns out that the social wasp and bees, which are the ones in the red, and I apologize for the font there, the ones in the red, in that red box, have fewer species. And it is known that in these social colonies, microbes are inherited from the pool of microbes in those communities themselves. Whereas the solitary wasps and bees get most of their microbes from the environment. Now, neutral models are great when they agree with things, but in fact, that's really not the most satisfying use of neutral models. The whole point of neutral models is that we actually think that neutral models are wrong, right? It's highly unlikely that the complexity of the microbiome variation and diversity can be explained by the simple processes that we define in our neutral models. And so it is actually nice when we can reject our neutral models, because they point to other processes that need to be added.
We do have to stop.
Right. And so I should just say that in terms of sponges, we have these two groups of sponges, one that has high microbial abundance, one that has low microbial abundance, and they actually run counter to the predictions of our neutral theory. And we think that, therefore, it allows us to talk about population subdivision and selection. And so these are our conclusions. All models are wrong, but some models are useful. We think that our neutral models are useful null models, and I shouldn't take credit for that quote. It comes from Box and Draper. Rejection of our neutral models allows us to make conjectures about additional evolutionary drivers, and I'll stop there. Thank you.
Thank you very much.